to express my gratitude for being invited here. Sometimes I'm not positive that I'm in the right place because I do something different. But be that as it may, I would really like to talk to you a little bit about collagen and how glycosaminoglycans affect them. Now this is a histology of normal skin, just for sake of review. This is the epidermis. The basal layer here is where the cells grow. They migrate up and become stratified, they lose their nuclei. And then they become the stratum corneum, which is what you present to the world. Now, down here is the dermis, and the dermis is responsible for structural features. Here's some elastic tissue, which is stained. Most of it, about 70% or so of this is collagen. So collagen is very important. But also there's an extracellular matrix, which is composed of glycosaminoglycans, and it's composed of hyaluronic acid, proteoglycans, and so on. And we'll go into that a little bit. Let's see. Uh-oh, did I go the wrong way? Yeah. Uh-oh. Wait a minute. What? Let's
see. Is it the photo? Try that one.
I did.
Yeah, there you go.
Oh, got the magic touch. Okay. Well, anyway, dermal collagens. These are, they say, the structural component of skin. They comprise 70–80% of the dermis. And the collagens are mostly type 1 and type 3 in humans. Of course, there are something like 23 or 4 collagens that have been discovered since then, but we're worried about mainly type 1 collagen. So, as we just said, they belong to a genetically distinct group of structural proteins. Type 3 and type 1 and type 3 are about 85 to 15, and we're focusing in this work on type 1. So structural aspect is tropocollagen is the basic unit of skin collagen. It consists of 2 alpha-1 chains and 1 alpha-2 chains, and these chains have a left-handed helical structure wound in a right-handed superhelix. It has a general formula XY gly, and it contains about 10 or 15% each of proteins and hydroxyproteins. And of course, they contain other proteins too, but these are the main ones. So we start out with tropocollagen, which is what I just described, a triple helical structure, and they form fibrils by linking with other tropocollagen molecules. And these are so-called normal linkages, but fundamental linkage involves formation of a shift base between free amino groups of lysine or hydroxylysine and nearby aldehydes, which are formed by lysyl oxidase on hydroxylysine residues. And this is a schematic representation, which I stole from an earlier edition of Fitzpatrick. And here's the shift base. This is not stable, but further reactions occur and make it stable. And so you get the cross-linking. And here's what it looks like under electron microscope. I'm sure you've all seen pictures of these. And what we have here, there's a tropocollagen. Then it's aligned in a kind of a quarter stagger configuration, and it forms these cross striations. It says 640 angstroms or 64 nanometers. Actually, we know now it's 67 nanometers because these were photographed at a kind of a funny angle, so it makes it look like it was 64 nanometers. And here's, just to belabor the point, here's tropocollagen molecule, the secondary structure. Here you can see the the right-handed helix, left-handed helix, excuse me, going this way. And here you can see the left-handed triple helix and the right-handed super helix. And this is what it looks like when you look from upside down, so that you don't get the idea that there's nothing between those. There actually is quite crowded there. Now, one thing that is not always appreciated is that these collagen molecules have non-helical regions. They're short N and C terminal ends, and these are called talopeptides. And these have the configuration of an anti-parallel beta-pleated sheet. Now, in contrast with a triple helix, these talopeptides have tyrosine, phenylalanine residues. These are hydrophobic. These are low in arginine and hydroxylysine residues and lysine residues. These are in a favorable position to form dimers or interact and form higher oxidation products. And these, what the function of these are mainly necessary for fibrogenesis, which we just saw. This is a picture, I guess it's a cartoon, you could call it, of telopeptide. Here's the the C chromophore. Yeah, it starts at the N protein, and it goes around in a kind of a semicircular way. But you see that the aspartic acid, glycine, serine, these are hydrophilic amino acids, and these are facing out towards the solvent. And all the tyrosine residues, the threonine, isoleucine, leucine, valine, these are all hydrophobic, and they're facing inside. That may be important. Now, why all this lecturing? Well, tyrosine can be degraded. Actually, it turns out it's a very active molecule. It can be degraded either by oxidation or UV radiation. And the interesting thing about this is collagen actually is fluorescent. And what you see mostly is tyrosine and or modified tyrosine. In other words, oxidized or otherwise degraded or UV radiated proteins. And they have different protein fluorescence properties than normal collagen. So why is this important? Well, one thing that we discovered is that these residues are photolabile. If you shine UV radiation on them, the fluorescence fades away. That means there's photochemistry, and it means they're forming non-fluorescent products. And so this raises a very interesting question. Well, what are these collagens like after the photochemistry? What happens? Well, we know that look at the skin commercials, you see that there's wrinkling of skin, among other things. And this is because of collagen. This is basically like a wound healing process. The collagen is changed somehow, and it stimulates MMP1, I think, or MMP2, collagenase or gelatinase, and these clear it out, and you have a matrix formed, and you got new skin. And this is how the body treats it. So why does photolysed collagen act as a foreign protein? And this is the question that got my attention some years ago. But now, here is something else we discovered almost by serendipity. This is the normal collagen spectrum. It's mainly tyrosine. although if you look carefully, you can find a little bit of oxidation here. This is a new sample, we had a sample that we inadvertently left in the refrigerator in the dark for about 5 years, and we were trying to see if there were any changes, and you can see there were. The tyrosine peak goes down, and you get these oxidation products, and they go up. So, now what about these fluoroforms in aged collagen? Well, you have tyrosine, which is the parent compound. Fluoresces at 305. You have dihydroxyphenylalanine, sites at 285 and bits at 330. Then you have dityrosine. Dityrosine is a covalent dimer of tyrosine, and it fluoresces at a much longer wavelength, 400 nanometers. And you have these other products, dopa oxidation products, that can excite roughly 350 nanometers and fluoresce roughly 450 nanometers. There are a whole bunch of them. And now we have, just for the sake of completion, we have these AGEs, advanced glycation products, which have these features. They fluoresce at about 450 nanometers or 440. And we're not studying these because these are reaction products between lysine residues and sugars, glucose or ribose or so on. And I neglected to point this out. This is another type of collagen oxidation product, dopa oxidation product, and unfortunately, we really don't know what it is. We've got some ideas, but what happens is let's go back. What happens is these oxidation products, they have second order, they're consistent with second order reaction between two like polymers. So that sounds like there's a double molecule somehow, and that's fading 2 molecules at a time. And it gives a nice fit to a 1 over F versus, actually 1 over C versus time. And then we have dimerization, and we'll go into that in a minute too, photodimerization. Well, we're talking about tyrosine dimerization to dityrosine. We found that there was a kind of a yin-yang relationship. Now we have fresh collagen, which is this, and we're plotting fluorescence versus time, and this gives a nice linear plot. And this is your aged collagen sample, the one that we looked at before that was in the refrigerator for 5 years. And what happens with this is that actually you have several processes. You have excitation with UV radiation, and you have a rate constant for that. You have the excited state, which is a tyrosyl residue, going back to the ground state, giving off heat. You have another reaction where it's reacting with another tyrosine molecule, and this is actually the slow step because the tyrosines have to diffuse together to dimerize. And so reaction 3 is the slow step in photodimerization, and it has to build up. When it builds up, actually it'll eventually bend over and form a steady state. But what we're doing is taking the reaction right at the beginning where this excited state is very small. So, in fact, as T is approaching zero, the rate of dityrosine formation is maximum. It's roughly equal to Ka because A star is much less than A. So that's how we get the kinetic curve that we got. Now we have, as I said, we have a photooxidation product, and this is, I guess you could call this the, whoops. This is what we could call the yang, where now this is the age sample, and now this is plotted versus 1 over F, which is a second order reaction. And you can see the black dot, the age collagen fades much faster than the so-called new collagen sample. So in that case, as we said, it's proportional to 1 over the product. Well, I guess now we've got the question, what does the surrounding extracellular matrix have an effect on tyrosine spectroscopy or photochemistry? Does it have any effect at all? I'm going to skip through this fairly quickly because you people know much more about this than I do. But matrix protein, you've got hyaluronic acid, which is the major component, sulfated GAGs, proteoglycans, elastins, and you've got other things which are, I guess, not relevant for this particular discussion. And here are just an idea. You can see in the dermis, most of the uronic acid as uronic equivalents per milligram of wet skin. Most of that is in the dermis. There's a little bit in the epidermis. We're not worried about that so much. Proteoglycan aggregate, we've seen this in a previous slide. This is the hyaluronic acid. This is linker protein. This is the protein core. These are GAGs, keratin sulfate. I think there's chondroitin sulfate there, and these are the structures, which I'm sure you all know. And we've seen this one already. I have to thank Ree for the lead-in. So the reason what we're doing is trying to approach this stepwise. And we haven't gotten very far, but what the first approximation to an in vivo situation would be hyaluronic acid and collagen mixtures. And, so we're not taking effective proteoglycans. Hopefully we'll be able to do that someday if I ever get funded again. But there are reasons to suspect that there may be some sort of interaction between HA and collagen mixtures. Degorski et al. Studied these interactions. They studied it by measurements of polyacrylamide gels so they could get the interaction closer together. And they concluded that these interactions are mostly hydrophobic, but interactions among hyaluronic acid molecules are ionic in nature. And they did this in the separate solutions. These are controls. Solutions, and they did it also in the mixture, and their results indicated either a hydrophobic or an attractive interaction between the segments of the two biopolymers. These workers favor the latter theory. I'm not totally sure if that's the whole story or not, but we still want to know, so does the surrounding extracellular matrix have an effect on tyrosine spectroscopy or photochemistry? It turns out, to make a long story short, and this is, believe it or not, this is the result of about 3 years worth of trying. This is an Arrhenius plot, 1 over t versus the log of the slope, which is related to rate constant. And you can see there's a lot of scatter among these dots. But if you look at 95% confidence level, there really is not much of any difference between those two sets of collagen over all these temperatures. This is starting at about 8 or 9 degrees Celsius, and this is about 60 degrees Celsius. And you can see that it's not straight, but that's because that reflects the helix coil transition. But even there, it doesn't seem to make any difference. Hyaluronate has variable effect on photooxidized dopa derivative. And you can see, in this case, the effect is small, especially down here at low times. When you get up to 100 minutes of photostation, the old age collagen starts to tear off, and this one's going to eventually do the same thing. You could say, well, in this experiment, hyaluronic acid seems to facilitate fluorescence fading after extended photolysis, but the effect is probably not significant, and that could just as easily reflect back reactions or other types of reactions besides equilibrium. Now, here's another slide which shows the opposite effect. Here is the aged sample. Here is the new sample. And from what we get, when you put all these results together, it seems like the effect is not significant. There may be a small effect on dityrosine formation. In this case, the buildup is slightly retarded by hyaluronic acid. It may or may not be significant. This seems to be more. This seems to happen more frequently. And here's another case where we got basically the same thing. So the jury is still out on that. So what we want to know is why. Well, for one thing, God damn it, hyaluronic acid is more viscous than water. It could lower the rate of dimerization since the two tyrosine molecules must diffuse towards each other. And based on previous studies, one might expect that there would be interactions between collagen and hyaluronic acid. The Nagorski et al. Indicated that was either hydrophobic or an attractive interaction between the two biopolymers. Well, but we don't really see much. And why could this be? Well, the photochemical intermediate is a tyrosyl radical. It's a free radical. These may not be so sensitive to ionic processes, and this favors the hydrophobic process. And tyrosyl residues are also in the hydrophobic region of the collagen molecule and not readily accessible to hyaluronic acid, so they may not interact because of that. So, in conclusion, hyaluronate has a small, if any, effect on spectroscopy and photochemistry of collagen fluorophores. We don't know the reasons for this, but we hypothesize that hydrophobic tyrosine residues in the talopeptide may be shielded from the extracellular matrix. And I'd like to just acknowledge grant support from that. And these are basically, this was a technician I had, and these were students. And thank you for putting up with me.
Maybe I have time for like one question from our person who dominates the questions here. So,
Julian, this is an important study in terms of sunscreen. Because you get UV radiation from the sun, we have sunscreen. How do you see the sunscreen interacting with the collagen in the skin in order to protect us from the radiation?
That's an interesting question. In this case, the sunscreen would act really as a shield, kind of a roof, and it would keep the radiation out. It doesn't, most of it doesn't penetrate, well, all of it doesn't penetrate into the dermis, but it's above most of the collagen. It's kind of layered there. Yeah, there's another question?
Yeah.
Sorry, thanks. This might be a naive question. This is not my field, but could you comment on the role of oxygen-free radicals like nitric oxide, if it has a direct interaction with the collagen? It has a reaction with the tyrosine. I mean, it should affect the spectroscopy. It has a role in healing. But is that understood at this point?
Well, we haven't studied that directly, but we have studied quite a bit the reaction between melanin, which is made from tyrosine, and nitric oxide. And nitric oxide gets oxidized on melanin, and actually in the process, oxygen gets reduced to superoxide. So you got these nasty molecules that are generated from the interaction between melanin and nitric oxide. And we got interested in that study because African Americans particularly suffer from keloids, and I thought maybe this was a reason why.
Okay, if there's nothing else, let's thank Julian again for a nice talk.